Genetic information is passed through DNA. In viruses, it's RNA sometimes. Prokaryotes typically have circular chromosomes, whereas eukaryotes have multiple linear chromosomes. And base pairing is conserved. So this whole idea of base pairing gets us into the idea of nucleotides. So we have the letters of the code. And purines have a double ring structure in their complex nitrogenous bases, whereas the pyrimidines have single ring structures in their nitrogenous bases. So what that comes into when we talk about base pairing is that purines pair with pyrimidines. So cytosine and guanine, they bond together through hydrogen bonds because they have three places where they are hooking together, whereas adenine and thymine only have two hydrogen bonds between them. So it's these hydrogen bonds that hold the two strands of DNA together. Now, when we say that they are conserved, and base pairing is conserved through all forms of life, so A and T bond together and G and C bond together, and those particular nucleotides are found in all forms of life, from bacteria to fungus to corn to animals. So we also have RNA, and the big difference is that they have a different sugar. Instead of DNA with deoxyribose, RNA has ribose. Typically, RNA is single-stranded, and instead of thymine, it has uracil. Now, RNA gets us into viruses because when we talk about viruses, they typically have some kind of a protein coat, maybe some enzymes, but they have RNA and sometimes DNA as their genetic material. So you can have spherical capsids. You can also have a helical capsid, and again, this one also has RNA as the heritable gene information. So we can also have this polyhedral capsid and a polyhedral head with a helical tail in the case of bacteriophages. So the thing about viruses is that they don't have the ability to independently use energy to offset the random breakdown of nature, respond to stimuli, or maintain homeostasis. So we don't really consider them alive, but they do reproduce and they do pass on genetic information from one generation to the next, but they need a host. So in this case, the bacteriophage is injecting the genetic information into its bacteria host, which then hijacks its machinery to make more and more copies of itself until it destroys the host cell and goes and infects more. Now, in addition to hijacking and making copies of itself, sometimes its DNA can be integrated into the host as well and get passed on through the next generations of binary fission of the bacteria host. We also have retroviruses like HIV, which can attach to eukaryotes, inject their genetic material, in this case it's RNA, and through reverse transcriptase can be turned back into DNA, be put into the host cell, and then as this DNA is transcribed, it actually makes all of the proteins and recopies the RNA that is needed to make the next virus, retrovirus. So when we get into prokaryotes, we think of circular chromosomes. We also typically think of plasmids. Plasmids are typically with prokaryotes like bacteria. Some archaebacteria contain plasmids, and some eukaryotes like yeast. But plasmids, we also often consider them DNA that is non-essential, but may give some special traits to that bacteria. They can also sometimes be picked up from the environment or transferred from one bacteria to another. So when we think about this, we don't have a nucleus in a prokaryote, but we do have this long circular DNA all kind of combined up, and then we have these little plasmids that can exist. And again, when we get into biotechnology, we'll talk a lot about plasmids. So again, as we go through this process, the information is getting passed on from generation to generation. As they divide, they have to replicate the DNA every single time through prokaryotes. And it's the same thing with eukaryotes. So when we're talking eukaryotes, instead of a circular chromosome, we talk about linear chromosomes. And we can see that when we look at karyotypes, because it's not one long ring, it's distinct individual strands of DNA. And again, we have our DNA stored in the nucleus as chromatin. It'll loosen up so then it can be read for transcription and translation. But ultimately, through mitosis and meiosis, we can pass that DNA and that heritable information to the next generation. So genetic information that's passed on is DNA, and sometimes it's RNA. Prokaryotes have circular chromosomes, eukaryotes have multiple linear chromosomes, and base pairing is conserved.